Hey, thank you, Arjun. I'll tell you about spatial omics of the intestinal epithelium. In my lab, we seek to understand how cells work together within the context of tissues to collectively achieve physiological goals. And one of our favorite organs is the small intestine. It has a beautiful shape. Our gut is lined with a single layer of epithelial cells that form these highly folded structures composed of mountain-like structures called villi. They're about 0.5 millimeter in height, and deep invaginations are called crypts. And as you can see by this movie from Hans Severs, at the bottom of the crypts, we have stem cells and progenitors that continuously divide each and every day of our life, creating a mitotic pressure that constantly pushes the cells upwards. So cells here are on the move. As they exit the crypt, they differentiate into two main cell types, secretory cells or enterocytes, the nutrient-absorbing cells. And then these cells rapidly migrate up along this axis at one cell position per hour. And for the next 4 days, they will absorb nutrients and fight bacteria until they reach the tip, and then they will be shed off. So we shed off massive amounts of cells, about 30 grams of cells every day. It's a quarter of our feces. And what we really like about this tissue is that space equals time here. So the higher up you are along this axis, the more time has elapsed since you were born from the crypt. And that's a very nice space-time equivalence that I'll talk about in this talk. Okay, so the villus is not a uniform structure. It's a highly graded structure. There are very high levels of oxygen at the bottom, and as blood flows upwards and cells respire, oxygen concentrations go down dramatically to even hypoxic levels at the tip. On the luminal side, we have inverse gradients of nutrients and bacteria. The luminal content flows in from the tip here, and as it flows downwards through reaction, diffusion, convection, the levels of nutrients and bacteria decrease towards the bottom of the villus. And then we also have morphogenetic gradients, high BMP levels at the tip, high canonical wind signals at the bottom. So it's a very graded structure. And we wanted to ask whether, in response to this graded microenvironment, different enterocytes, these nutrient-absorbing cells, specialize in distinct functions at different villus heights. And when I say we, I mean my previous excellent postdoc, Andreas Mohr, who opened his lab in ETH Zurich lately. And so what Andreas did was to section the gut and use laser capture microdissection to catapult 5 different zones of the epithelium from the bottom of the villus to the tip into 5 different tubes. He performed bulk transcriptomics, and he found quite a high transcriptional diversity. So we found a bunch of genes that go down in the epithelium from the bottom to the tip of the villus, and some genes that go up, and we call these landmark genes. And we then use them to localize single sequence cells to infer where they were sampled from. So we take dissociated epithelium, and for every enterocyte, we examine the combined expression of these landmark genes, and we retrospectively infer what was the coordinate along the villus axis that they came from. And then we can group together these spatially stratified enterocytes, average their gene expression, and get a complete transcriptional gene expression map, which is what you see here on the left. Every row is one of the enterocyte genes, and the different columns are the different zones. So the left column is the crypt, and then we have the bottom of the villus, the middle, the tip. And you can see that transcriptionally, these cells are very, very diverse. And you could say, of course, crypt would be different than the villus because crypt contains the proliferative cells, and the villi contains the differentiated cells. But even if you erase the crypt, you can see that there's extreme diversity. And if you take a tip enterocyte and a bottom enterocyte, they're as different as an enterocyte and a secretory intestinal cell. So completely different cell states. In this fish validation, you can see 3 examples of zonated genes. As the enterocytes exit the crypt over their first day of life, they express the green gene, then they shut down the green gene, upregulate the red gene, shut down the red gene, upregulate the blue gene, and then they're gone from the tip. I'll tell you just in a few slides about some of these zonated functions because this is published work, and then I'll move to the non-published part. So as the enterocytes exit the crypt, the first thing they do, they don't engage in absorption. They actually fight bacteria. They express antimicrobial peptides that are secreted into the lumen and presumably protect the crypt from bacteria. Remember, the crypt is the holy grail of this tissue where the stem cells reside and should be viable for the organism's lifetime. There's also NLRP6 that encodes the inflammasome bacterial sensor in this zone, and a bunch of cytokines that are secreted into the basal side and specifically recruit some immune cell types into this bottom villus zone. Then as the enterocytes migrate upwards, they sequentially express the machinery for absorption of amino acids, then carbohydrates, then peptides, and at the very tip, apolipoproteins and cholesterol. And at the very tip, there's hundreds of genes that are upregulated in enterocytes just hours before they're shed off, including some key transcription factors, EGF receptor, and a very interesting extracellular protein program centered around this gene called NT5E or CD73. These are a bunch of genes that take up ATP, convert it to AMP, adenosine, and then suck up the adenosine. And all of these genes are expressed at the tip, and we think they're creating a luminal microenvironment that's depleted of ATP. Bacteria secrete a lot of ATP, and this is a danger signal for the immune system. There's a lot of immune cells here in the stroma, so there's an opening here in the tissue, and to reduce inflammation and attack by these immune cells, we think the enterocytes are reducing the ATP concentration as a form of immune modulation. Okay, so just to summarize this part, the previous picture we had of the gut consisted of stem cells, progenitors, and then what we call ternally differentiated cells. And what we think we've shown is that enterocytes are actually not ternally differentiated. They're constantly trans-differentiating. They're starting off as these sterilizing enterocytes, then switching to amino acid absorption, carbohydrate absorption, lipid absorption at the very tip, this immune modulation. Okay, so now I'm going to switch to my second part, and I'll tell you about a question I always get whenever I give this talk, and that's the question all of us fear. What about the proteins? So I freak out when I get this question, as I'm sure many of you, because measuring proteins is very hard. But in this tissue, I want to convince you that measuring proteins is actually very important. So RNA and proteins don't always correlate because there's post-transcriptional regulation consisting of variable translation, variable protein lifetimes. But even if these regulatory rates are constant, you can have very interesting dynamics in this specific tissue. And this is because the gut, the villus, is a system that is not under steady state. If you remember, the migration time from the bottom of the villus to the top is 4 days. That's extremely fast. That's about the order of the magnitude of a typical protein lifetime, which is about 2 days. And this means you can get some very interesting out of steady state dynamics. So let's imagine our villus axis here, and this is time, but remember space equals time. And we're plotting the RNA in blue and the protein in red. If I have an unstable protein, it will closely track the levels of the RNA, right? So RNA would go up and then down, and protein will only also go up and then down. If I would sample cells along the spatial villus axis and measure both RNA and proteins, I will see a correlated behavior. So as RNA goes down, also protein goes down. On the other hand, if we have a very stable protein, we could see a very different behavior. Now RNA would decrease as the enterocytes migrate upwards, but the protein will continue to accumulate because of this for like 3 or 4 days. And if we sample single cells along our villus axis, we would actually see an anti-correlation. So these are different cells along in different positions. As the RNA goes down towards the tip, protein levels actually go up. So you can get very interesting dynamics that depend on the protein lifetime. So we want to measure, get the same whole genome map of both the proteins and the RNA. And Yotam Harnik, an amazing PhD student in the lab, decided to take on this challenge of obtaining these global protein measurements with high spatial resolution. And he decided to use a technique that we called spatial sorting. So the idea here is that we already have a transcriptional atlas of enterocytes along the villus axis. We're going to look for surface proteins that are zonated. And we're going to utilize them to isolate bulk populations that are spatially stratified. So I'll give you an example here. Let's say that we have the green surface marker that's only expressed in enterocytes in the crypt, and a yellow surface marker that's expressed in enterocytes in increasing levels towards the tip, and then a red surface marker that's only expressed in enterocytes at the tip. We can then dissociate the enterocytes, incubate them with a cocktail of fluorescently labeled antibodies against these 3 markers, and then we can use the green surface marker to negatively select for the crypt and retain the villus cells. And we can examine the yellow-red fluorescence, and the cells with high red, high yellow would be the ones at the tip. Cells with no red and little yellow would be the enterocytes at the bottom, and intermediate populations in these FACS plots would be the intermediate zone. This will allow us to gauge these populations, sort hundreds of thousands of cells, and then we can perform any omics measurements we would like to perform. So for example, bulk RNA sequencing, mass spectrometry, proteomics to measure proteins, attack-seq, different epigenetics measurements, etc. And this is inspired by a protocol developed previously in the liver by Shani Ben Moshe and Jonathan Shafir. Okay, so Yotam converged on these 3 surface proteins. CD24 is only expressed in the crypt, CD26 expressed in an increasing manner from the bottom of the villus to the tip, and CD73 that's only expressed at the very tip enterocyte. And then he associated the enterocytes, incubated them with these antibodies, and this is how the fat spot looks like. You see the correlated increase in CD73 and CD26. From each one of these gates, he sorted bulk enterocytes, measured RNA, and we compared the bulk RNA sequencing measurement with our previous single-cell spatial reconstruction. So blue is the spatial sorting, bulk RNA sequencing, and yellow is the single-cell spatial reconstruction. You can see we have a very nice overlap. So we believe that we're sampling indeed these spatially stratified enterocytes. Now Yotam performed both RNA and protein measurements. And he got a very nice deep measurement of more than 3,000 proteins that allowed us to build this protein zonation map with different functions allocated in different zones. But I don't want to stall on this slide because I want to immediately go to the RNA protein relations, which I think is the more interesting part of this story. So here you can see the zonation profiles of both the RNA and protein for some genes. RNA in blue, protein in red, from the bottom of the villus to the top. And you can see that there's a very nice correlation for some of these genes, like RPL27 and ribosomal protein. Ribosomal proteins go down at both the RNA and protein level. REC3B, this defensin antimicrobial peptide secreted from the bottom of the villus. It's zonated towards the bottom of the villus, both at the RNA and protein level. And these TIP enterocyte program, immunomodulatory programs, also show correlated RNA and protein behavior. But for many, many genes, we saw an anti-correlated behavior. So you can just look at NLRP6 here, the inflammasome gene as an example, where the RNA goes down from the bottom of the villus towards the tip, whereas the protein actually goes up. So if I just look at the RNA, I would say the gene activity is higher, inflammasome activity is higher at the bottom of the villus, but the protein tells us a completely different story. And this is a very broad result. I'll just show you another visual demonstration. This is the gene STAC5A1 that encodes SGLT1, the main glucose luminal transporter. At the RNA level, you can see that it's zonated towards the bottom or the middle of the villus, and it's very low RNA levels at the tip. But when you look at the proteins, you see a completely different picture where the protein levels increase towards the tip. So the zone where you have high protein expression is where you have low RNA expression, and vice versa. The zone where you have low protein expression, you have high RNA expression. About 40% of enterocyte genes are anti-correlated, are spatially anti-correlated along the villus, and this is very different than the liver, where the majority of genes show perfect spatial correlation between RNA and protein. Okay, and this goes beyond individual genes. Whole processes are zonated differentially: fatty acid metabolism, peroxisomes, oxidative phosphorylation. This is just an example of respiration. If I look at the RNA for genes that encode the respiratory machinery, I would say that as the enterocyte migrates upwards, they respire less. Makes sense, they have less oxygen. But when you look at the protein, a completely different story, they actually respire more. And having this protein map was very important for us to revise the functional map along the villus axis. Okay, now I'm going to switch to mechanisms. What gives rise to this profoundly different zonation patterns of RNA and protein? And we know that RNA, denoted here by M, makes proteins through the process of translation, and we know that proteins are degraded or secreted. And both of these translation or secretion rates could depend on space. Remember, space and time here is equivalent, right? We have the differential equation. The change in protein levels at every coordinate along the villus is the translation rate times the instantaneous levels of the RNA, minus the degradation rate times the instantaneous levels of the protein. So we could have a situation where these rates are zonated, and for example, at the bottom of the villus, NLRP6 is translated inefficiently, but as enterocytes move upwards, translation efficiency increases in the tip. Or conversely, it could be that protein stability is higher for NLRP6 at the tip zone compared to the bottom zone. But we could also have this discordance just because of the out of steady state dynamics. And this is what we wanted to test. So we basically addressed this very, very simple model where, remember, we measure the spatial profiles of RNA and protein. And now we can plug in different translation and degradation rates, protein translation and protein degradation rates, solve this differential equation because we have the RNA profile, and get a protein profile and compare it to our real measured protein profile. And Lisa Buchauer, a very talented postdoc in the lab, has done this with a very nice Bayesian inference model, where basically she samples different combinations of translation and protein degradation rates and calculates the posterior probability of getting the measured protein profile. Okay, so black is the reconstructed model profile, and red is the measured protein profile. And you can see that with these uniform rates, we can explain many of these discordances just simply because of variable lifetimes. For example, this H2K1 has a short protein lifetime, so it tracks the protein tracks the RNA level. GSA1 has long lifetime. It acts like an integrator. RNA goes down, but protein continues to accumulate. And practically most of the genes in enterocytes can be explained by this space-independent model, which if you think about is very, very strange, because it means that in this tissue, RNA determines protein levels, but not in the same place and not in the same time. So a gene that's expressed at the bottom of the villus at the RNA level determines the protein levels that are needed at the villus tip, 0.5 millimeter away and 3 days away, which is a little bit strange if you think about it. Okay, so in the last part of my talk, I want to ask whether there is some kind of underlying design principle here in having so many genes where the RNA is expressed at the bottom of the villus or the middle of the villus, and the protein is expressed at the tip. And I want to go back to my first slide and remind you that this villus axis is highly zonated. At the bottom of the villus, we have high levels of oxygen. At the tip, we're in nearly hypoxic levels of oxygen. It's about 8-fold decline in oxygen concentration. When cells have a lot of oxygen, they have a lot of ATP availability. So this is the energetically rich zone, the bottom of the villus. On the other hand, the business end of this tissue is, at least in terms of absorption, is the villus tip, because this is where most of the nutrients are. And in order to efficiently absorb nutrients, the nutrients absorption machinery needs to operate very efficiently at the tip. And we know the gut is super efficient in absorbing 90-something% of the nutrients that go into it. Okay, so we have machines, the transporters that bring in the nutrients, and it takes energy to make them. Translation takes energy, and also takes energy to operate them, because you have to pump in nutrients against the concentration gradient. It just takes too much energy in a zone that is compromised for energy because there's not a lot of oxygen. So if you can split temporally or spatially split the two processes of making the machines at the bottom of the villus and then having them ready at the tip, you could optimize the function of these machines, these transporters at the tip, because you're going to have more available energy. You're not going to need to waste energy on translating the proteins de novo. Just to show you that this is the case, if we look at the protein levels of both the ribosome and the total RNA levels in single cells along the axis, which is the substrate for the ribosome, both of these go down. So the bottom of the villus is the site of massive translation. And when we look at the transporter activity or intestinal absorption protein, the protein activity, the protein amount go up towards the tip. So there's this spatial separation where protein production is at the bottom of the villus, nutrient absorption at the tip, and we believe this optimizes overall tissue function in face of these constraints of low oxygen availability and high nutrient availability at the tip. Yeah, we think we think this could explain the sort of paradoxical finding that crypt cells divide like crazy. Okay, they need to divide like crazy if we want to maintain this 4-day migration rate, and having this 4-day migration rate is the only way where you can separate these two processes. If it would have taken 20 days to migrate from the bottom of the villus to the tip, I would not be able to make proteins at the bottom and have them survive until the enterocytes reach the tip. So in order to have the separation, you need to have high migration rate that's on the order of the protein degradation rate. So I'll summarize. I've shown you spatial sorting that enables reconstruction of omics maps of the villus. I've shown you proteomics maps. We can also reconstruct the microRNA, ataxic, and other omics features. Many genes have anti-correlated spatial RNA protein profiles in enterocytes, surprisingly. Translation of stable proteins at the villus bottom reduces load for the resource-limited upper villus zone. And we think the strategy of spatially or temporally separating protein production and protein activity could be a general design principle for other short-lived lineages, for example, neutrophils, erythrocytes, skin cells, and other tissues. So I'll end here, and I'll just thank Yotam Harnik, the PhD student who did most of the work together with Lisa Buchauer and Andreas Moore, who did the first part, and funding sources. And we are recruiting, if you're interested in these or other projects related to tissue biology, please contact. Thank you.
Thank you very much, Shalit, for the wonderful talk. Got a bunch of questions here. So first is Reagan Hamill, who says, really interesting data. Do you think there's any way of predicting the type of correlation between mRNA and proteins from an ex vivo assay like transcriptomic data? So I guess if you lose all the spatial information, if I'm interpreting this right, is there still some way to predict the type of correlation you might see?
Okay, I'm not sure. So there are methods, independent methods that have measured protein lifetime and protein translation rates, and we did correlate. We sort of we infer these processes from the spatial separation of RNA and proteins in the tissue. There's a very nice paper from Gunnar Hansen that gavaged the amino acid, metabolically labeled amino acid, and then measured Bach proteomics and sequential days, and we have a nice correlation with that data set. There's ribosome profiling that we and others have done, and our estimates for translation rates correlate with that. So yeah, I think, I'm not sure I get the question, but there are independent methods for measuring these rates. Yeah, and many of them have their limitations.
Yeah, I think that's sort of how I interpreted that question as well. One other question that I have perhaps related to this is the inference of the prediction requires knowing like the flux along that spatial axis. So is that something that you have like a hard measurement for, or is it sort of like a fit parameter?
Yes, it's a great question. So this is sort of known, and it's about 1 cell position per hour. It's dictated by the amount of proliferation in the crypt. So the more cells divide in the crypt, and the faster their cell cycle, the faster the migration rate. So basically, this is mitotic pressure, and it's been measured by lineage tracing and EDU incorporation, and then measuring where the cloud of labeled cells migrate at different times. Very interestingly, I can say it's very different than human. Human, there's less dividing cells, and the villi are longer, and the time scales become interesting in human. We started to look into that.
Huh, that's cool. Okay, let me go to another question here. So, from Tejasav Sarawat at the Mayo Clinic, considering heterogeneous transcriptome protein expression in different cells in the villus, have you considered understanding the effect one group of these cells exert on another group of these cells?
Okay.
Yeah.
That's interesting. If I interpret it correctly, so whether there's going to be protein heterogeneity within a zone, or maybe is that?
Yeah, I guess like if one cell type, I guess like how might they affect the expression in another one? So, okay. And then the rest of the question goes on to say, also keeping your liver zonation paper in mind, such an effect might be important for hepatocytes, which secrete many proteins. So I guess it's a question of communication.
Yes. Okay. So we really see. So first of all, we're measuring average protein expression because of this. That's an limitation of the spatial sorting. Now there's the emergence of single cell proteomics, which would be amazing when this could be applied to a tissue like the gut. I think we're going to start to see some heterogeneity within zones. The liver and the intestine, we've done spatial sorting. We've done a very similar study in the liver. Very, very different. In the liver, there's a very nice repeating anatomical unit with 12 layers, and there's no movement of cells. So we don't have this out-of-phase dynamic. And RNA largely dictates proteins. Also here, RNA largely dictates proteins, but in a very convoluted way that's transformed by these variable protein lifetime.
Yeah. So Sveta Morali asks, well, it says beautiful story, and asks, are similar patterns of expression, i.e., from crypt to tip, conserved in vitro organoids despite the sorry, these things are jumping around. Okay. Any ideas on how mRNA protein correlations behave during development or critical periods when the GI changes? For example, transitioning from infancy to more adult phenotypes.
It's a beautiful talk, and we have a rotation student looking at that right now. So there's sort of the pre-weaning phase, and then the post-weaning phase. During development, the mouse is born with complete villi, and then you have crypt morphogenesis right after birth. But there's profound changes in metabolism, right? It's like a many amino acid and fat diet before weaning, and then a carbohydrate diet that's more intermittent after weaning, and there's a lot of changes. So we're definitely very interested in that and looking into that, as well as the high-fat diets and different calorie restriction and different diets that give rise to profound changes, even in the length of the villi. So we think these patterns could change in an interesting way.
Cool. So Francesco, perhaps I'll end with one last question. So Francesco asked, are similar patterns of expression conserved in vitro organoids despite the morphological environmental differences?
Yes, so in mice, organoids don't make villi, and so it's impossible to test that. There's a human organoid system from Jason Spence and others that have more similar structure to the real human intestine, but we haven't looked at that. And of course, it's a great idea to look at that, and because in organoids you can really perturb the system and really measure the effects in much more detail. So it's a great idea.